The basic mechanism of inheritance was proposed by Gregor Mendel. He was monk, priest, in a monastery located in Austria. In a small garden of monastery, he bred different varieties of garden peas. Mendel selected pea plant, Pisum sativum, for his hybridization experiment. He carried out a large number of experiments for about 10 years, from 1854 to 1864. In Mendel's opinion, the organism selected for genetic experiments should have the following features. The plants are easy to cultivate. The reproductive structures are completely enclosed by the petals, so that the plants are normally self-pollinating. However, cross-pollination is also possible in them. Artificial crossbreeding is also possible, and the resulting hybrids are fertile. There are several traits with distinct characteristics available for study in pea plants. For example, tall and dwarf traits, etc. He observed these features are present in pea plants. Each trait studied in pea plants had two distinct forms. Mendel succeeded in his work not only because he selected the right organism for his experiments, but also because he analyzed the results by using the principles of statistics, like ratios. Mendel meticulously chose 7 contrasting characters in garden pea plants for his experiments. These traits included seed shape, seed color, flower position, pod shape, pod color, and stem length. Each of these traits had 2 different forms, which made them ideal for studying inheritance patterns. For instance, seed shape could be either round or wrinkled, while seed color could be yellow or green. Flower color varied between purple and white, and flower position was either axial or terminal. Pod shape was either inflated or constricted, and pod color was green or yellow. Lastly, stem length could either be tall or dwarf. By carefully observing how these traits were passed from one generation to the next, Mendel was able to formulate the foundational principles of inheritance, which we will explore in the next chapter. Mendel identified 7 contrasting characters in garden pea plants: seed shape, seed color, flower position, pod shape, pod color, and stem length. These traits were key to his experiments and helped him formulate the principles of inheritance. In the light of his experiments, Mendel proposed that there are special factors in organisms, which control the expression of traits and their transmission to the next generations. The discovery of several principles of inheritance by Mendel is the cornerstone of modern genetics, and Gregor Mendel is considered as the father of genetics. Dominant and recessive trait. When 2 contrasting traits are crossed and in the F1 generation, one of these contrasting characters expresses itself while the other does not. The expressed character is called dominant, while the other trait, which is not expressed in the F1 generation, is called recessive. First parental generation P1. The parents of the F1 generation are called the P1 generation. First filial generation F1. The offspring produced as a result of a cross between P1 is called F1. Second filial generation F2. The offspring of the F1 generation are called the F2 generation. Genotype. The genetic makeup of the character that is homozygous or heterozygous. Phenotype. The external appearance of the character, such as allele or gene, is called phenotype. Homozygous. Organisms carrying 2 identical alleles, for example RR or RR, are called homozygous. These homozygous traits are also known as pure breed. Heterozygous. Organisms bearing different alleles, for example RR, are called heterozygous. Monohybrid cross. A genetic cross between parents that differs in the alleles that possess for one particular gene, one parent having 2 dominant alleles and the other 2 recessives, is called a monohybrid cross. All the offspring, called monohybrids, have 1 dominant and 1 recessive allele for that gene. Crossing between these offspring yields a characteristic 3:1 monohybrid ratio in the following generation of dominant to recessive genotype. The monohybrid cross explains Mendel's law of segregation of genes, also known as Mendel's first law. Mendel's law of segregation, also known as the monohybrid cross. Mendel worked with pea plants and selected 7 pairs of contrasting characters. He got pure varieties of these traits. For example, one trait he studied was plant height. One pea plant was tall, while another was dwarf in height. Mendel crossed this pure breeder tall with pure breeder dwarf and got the F1 generation. In the F1 generation, all pea plants were tall. Mendel then allowed all F1 plants to self-pollinate and obtained the F2 generation. Mendel noticed a 3-to-1 ratio in the F2 generation. About 3/4 of F2 plants were tall, and about 1/4 of F2 plants were dwarf. From the result of these experiments, Mendel formulated the law of segregation, which states that allele pairs separate or segregate during gamete formation and randomly unite at fertilization. A dihybrid cross is a cross between two individuals that are heterozygous for two different traits. For example, in pea plants, seed color and seed shape are two different traits. The yellow seed color is dominant over green, and the round shape of the seed is dominant over wrinkled. When round and yellow-seeded pea plants were crossed with wrinkled and green-seeded pea plants in the F1 generation, all plants were round and yellow. When round yellow-seeded plants of the F1 generation were crossed and obtained the F2 generation. In the F2 generation, phenotypically 4 types of pea plants were obtained in the ratio of 9:3 to 3:1. The dihybrid cross explains Mendel's law of independent assortment, or Mendel's second law. Mendel's law of independent assortment. After monohybrid crosses between contrasting traits of pea plants, Mendel shifted his attention to dihybrid crosses. In one of his dihybrid crosses, he selected a homozygous pea plant that produces round yellow seeds, RRYY, and crossed it with a homozygous pea plant that produced wrinkled green seeds, RRI. In the F1 generation, he got all plants with round and yellow seeds, RYA. It proved that round and yellow were dominant characteristics. After getting the F1 generation, he self-crossed the plants of F1 and got the F2 generation. Out of 556 plants of the F2 generation, Mendel obtained 315 round and yellow, 108 round and green, 101 wrinkled yellow, and 32 wrinkled green-seeded plants. So in the F2 generation, besides the parental types, new combinations were also produced. These new types of plants, that is wrinkled yellow and round green, indicate that recombination of traits had occurred. Mendel noted that the offspring appeared in the F2 generation in a 9:3 to 3:1 ratio. These observations led to the formulation of Mendel's second law, that is, the law of independent assortment. This law states that when a pair of contrasting characters are crossed, the characters are inherited or assorted independently of each other. Comparison between the law of segregation and independent assortment. The law of segregation is a principle that describes the separation of the two copies of each hereditary factor during the production of gametes. The law of independent assortment is a principle that describes the independent assortment of alleles of different genes during the formation of gametes. The law of segregation uses a monohybrid cross, while the law of independent assortment uses a dihybrid cross. In the law of segregation, the ratio of offspring is 3:1. In the law of independent assortment, the ratio of offspring is 9:3 to 3:1.